All right.
Scott, you have to let me share.
Oh, I'm so sorry. Um, Make co-host.
Sweet.
There must be a way you can have it do this automatically.
Probably. But so this is like the interesting part of the Nature paper I got to read, where they actually identified and test a bunch of inhibitors for KRAS G12C mutants, and they do it via two different methods. So the first one is the disulfide tethering that I talked about in the last presentation, and then they switched to a different approach to making covalent bonds that are basically like unbreakable. So an overview is about like the primary tethering approach. So what we talked about in my last presentation, how they applied it to RAS instead of an E. coli enzyme from their original paper. The selection and modification of this final tethering compound. So they just kind of stopped there. They found the one that worked best. They edited some structures on it to make it work even better. And then they just kind of switched to something else, something other than disulfide tethering. And then there's a bunch of data where they test these inhibitors against RAS and see basically how effective they are against cancer. So an overview of what they did. They screened a library of 480 disulfide-containing small molecules. So that's a lot. But only 1.9% showed greater than 60% modification. And I didn't know what this means, but now I do. So 60%, that threshold of greater than 60% modification means that suppose you had 10 RAS proteins in a tube. This is obviously a teeny tiny number. And if you had 60% modification, 6 of them would bind to the compound. So they wanted more than 60% of the RAS molecules in this like grouping to have this small molecule bind to it. And only this many of them actually did that. So you can see the percent modification means what percentage of the RAS contained in that sample was able to be modified by these small molecules. And there's a clear winner down here, 6H05, which had 95% modification. So that's the one they chose. And they're thinking maybe we can make this better. So how they chose to like pursue this pathway of making this particular structure better is by judging the greatest degree of modification. So out of these, the very few that actually met the 60% threshold, it was 6H05. Then they wanted to see if it didn't react with the wild type KRAS, only the mutant. Because it wouldn't be helpful if you had an inhibitor that inhibited RAS that was functioning normally. You want it to inhibit the one that's non-functional or causing cancer. And they wanted to see if the compound did not decrease in the presence of GDP. So this is saying that it occupies an allosteric site instead of a competitive site. So GDP still needs to be bound to RAS. This compound has to bind as well. So it's saying that they have to be in two different pockets. So that's what they wanted to try to prove with this original tethering method. So they chose 6H05, and then they wanted to modify it. So what they did is, obviously 6H05 showed the highest percent modification at 95%, and they took these two, hold on, it'll make it easier. Where's my laser pointer? It's not letting me have one.
Oh, really?
And now it's making subtitles. Okay, now it's not. So
I'm
anyway.
glad you're using your laser pointer because people so rarely do, and I'm always internally saying to myself, "I wish you would use your laser pointer." So this is great, Beth.
It
You're modeling very good behavior.
Good to know. But they took these two like substructures within 6H05 and they tried to modify them a bit to see if it could make the percent modification any better. So this is that disulfide tail with the 2-carbon bridge between the amide and that linker, which is necessary to keep. And then they wanted to change like this little benzene derivative over here to see if they could make that any better as a binder. So here's 6H05, and they kind of grade it out. So the relative potency is 1, because it's the baseline. This just means how much better are these modifications than the original 6H05. So as you can see down here, 4.2, it's 4.2 times better. It is better at binding to RAS than the original. And this is called compound 6. So this is what it looks like if you put it all together, because it's kind of hard to tell when it's all broken apart like this. So this is what compound 6 looks like. So they really didn't change too much on this, this disulfide side, but they switched the sulfur over here for an oxygen and added an extra chlorine. So you can see like the changes in red. And these experiments basically were just useful in learning more about S2P, which is the switch 2 pocket. Ultimately, they didn't pursue tethering compounds for the remainder of the paper, but these experiments using the tethering helped them understand more about how small molecules, interact with the cysteine 12 within the molecule and how that secondary pocket works. So
can I say something real quick, Beth,
yeah.
about the changes? So yeah, that's true. On the benzene ring, all they did, yeah, they changed the sulfur to an oxygen and they added that extra chlorine. The other side, does everybody see that they flipped the amide versus 6HO5? So they just like so. And then they also made it a little bit longer. So they distanced the aromatic ring from the cysteine a little more, and that was helpful. So yeah, so anyway, so I think they definitely learned some stuff.
They learned a lot more about like the structure. So that helped them with their shift from disulfide compounds. So in the paper itself, it just said like 6 was the best tethering compound, but we don't want to do that anymore. So they wanted to pursue irreversible cysteine bonds. So they want covalent bonds. So what they did is they have these two starting structures of a vinyl sulfonamide. So here's that sulfonamide and then a double bond, and then acrylamides, which is that double bond there. And they wanted to make a covalent bond with them. And these look, if I go back, very similar to this kind of backbone here. The one exception being they added another nitrogen for the acrylamide compounds within the ring, up here I mean, instead of when it's spaced out here. That's like the one main difference. And then they just kind of varied the R groups. Within CC we have like an acetal here, extra chlorine, we added an iodine, which actually helped a lot. And what they wanted to do is that these electrophilic binding compounds, like I said in the previous presentation, disorder switch 1, cause a lack of coordination with the magnesium 2+, and displace glycine 60. So all three of these factors contribute to KRAS not being able to bind correctly to GTP, indicating that it's not able to be on all the time, which is what causes the issues. So these percentages down here just indicate that percent modification again. So these two kind of suck, 10 and 11, 14 and 28%. Obviously those weren't shown much throughout the rest of the paper. 7 is 50, 87, and then 9 and 12 are 100% modification, which is fantastic. It means that every RAS protein that was in that tube got bound to these Michael acceptors. So that conjugate addition of the thiol on the cysteine to this small molecule. And I took this from the paper. It's a bit disordered because it's 3 compounds laid on top of each other within RAS. So everything is all one color. So if you see like this light blue is compound 8 as well as RAS it's bound to. So you can kind of see how they're all interwoven. The white is 9 and then the dark blue is 11. And they're all like laid over each other so you can see how they kind of fit in. And then this right here, that yellow, that's the cysteine. So that's how it's bound. So you can see that there is a distinct link between small molecule and the protein itself. And then this is GDP shown off to the side. So you can also prove that it's 2 separate pockets. It's not disrupting GDP at all. It's still able to fit. And they divided these pockets into like the O and P sub-pockets. I'm not entirely sure why, but there's these 2 separate kind of alcoves within that allosteric site that these substituted benzene rings fit into perfectly.
So are they calling them that, like that's the pocket that's sort of at the ortho position versus the para position on the ring?
That is probably correct, because they didn't really say, and I just noticed that that notation does look similar, but they're just like, we called these O and P sub-pockets. Okay. That's probably right. But what I did, because this is a little, for me, this was kind of hard to see because everything's over top of each other. I couldn't see the structure of the compound. So what I did was I went into Chimera and I showed compound 9 bound so you can actually see how it fits within the structure.
Ooh.
So I was able to target one particular cysteine. Thank you, Scott, for teaching me that that's possible. And you can see that direct bond between compound 9, which had 100% modification, so I picked that one, and the cysteine. There's the magnesium ion, which is coordinated to GDP, which I left in all orange because it looked a little funny when it was all colored by heteroatom because they looked like the same thing. So there's GDP over in its own pocket. And then back here, you can't see there's the chlorine on the benzene. There's the iodine, that ether position. There's the amide link between the benzene, that nitrogen within the cyclohexane ring. There's the sulfonamide right there. And then there's that link between the sulfonamide and the cysteine itself. So this just goes to show that it does form an actual link within RAS, and it's not in the same space as GDP. This is just another schematic from the paper that shows why this is actually helpful, rather than just me like kind of talking about it. I am honestly not sure why they chose to compare HRAS to KRAS, because specifically this paper focused on inhibiting G12C mutations in KRAS as they are most prevalent, but the general idea remains the same. So here are those, I originally only mentioned 2 key amino acids, that threonine 35 and glycine 60. There's also tyrosine 32, which does hydrogen bond with the gamma phosphate on GTP. So this just shows that like loaded spring mechanism, which is why these are all like squiggly. So when GTP is bound, that third phosphate, it pulls these 3 amino acids in, which is what causes switch 1 and switch 2. And then when GDP is bound and that third phosphate is lost, you can see they kind of rotate out. And it's less of a linkage between them because there's no longer that third phosphate. These weird bubbles are where the third phosphate normally is. So it's showing the distance, how it increases. So here's threonine 35, and that distance is really pronounced in tyrosine 32. So here how short it is here when it has the gamma phosphate, and then when it's bound to GDP, how wide of a separation it is from where it should be. And then what they did here is they kind of superimposed compound 9 and they switched to KRAS. I really do not know why they chose to compare the two, but you can see how it disrupts even the distance between GDP bound. So it pushes those switches outward even further, causing disordering of switch 1, which is why it looks so funky in red out here, because the compound kind of covers up that cysteine and pushes switch 2 out, which subsequently pushes switch 1 further. So that's how these actually work. They're able to disorder the rest of the protein by introducing the smaller molecule into an allosteric site. So here's a lot of the supporting data as to why the compounds work and how they did these different tests to prove that they're actually functional against RAS. So they wanted to prove that compounds inhibit G12C KRAS exclusively, because what would the point be if it like targeted other cells in your body? That wouldn't be good. So compound 12, which had 100% modification, and it was one of the acrylamides instead of the vinyl sulfonamides, so we just kind of switched gears, was tested against obviously G12C KRAS and bovine serum albumin, BSA, which the reason they chose this is because it has 1 free cysteine as well. That just means it's like able to bind to. And so this is protein mass spectrometry. That is what this shows. And this is just like regular mass spec, except it's huge because it's proteins. So this is the mass in Daltons of there's bovine serum albumin, and there's KRAS plus 12. So what this is showing is that this compound, this molecule, the bovine serum albumin, the BSA, did not bind to 12, or 12 did not bind to it because it's the one doing the binding. KRAS did, and it bound 100% of the time. So this is showing that the mass, obviously, of KRAS increased, not by much comparatively, because it has the small molecule bound to it, and it did not bind to BSA. Even though it has a cysteine available to bind, it targets specifically this mutant. And they wanted to test that even further. They're like, okay, so it doesn't bind to another enzyme, another enzyme with a free cysteine, but what if it binds to wild-type KRAS? What if it inhibits regularly functional KRAS? So they tested it against wild-type KRAS. And this just shows the percent modification comparison instead of the actual protein mass spec comparison. But 100% of the time it bound to the mutant, And 0% of the time with tiny error bar, it bound to wild type. So this proves that A, it only targets KRAS, and B, it only targets mutated KRAS G12C. That's good. And then we wanted to see, does it actually do its job? Does it keep GDP locked in and not exchange it for GTP? So this is kind of interesting. This is kind of a comic that shows how these experiments were carried out. MANT is just this group they added to GDP to make it fluoresce. So you can kind of tell where it is if you do these absorbance, like what we did kind of in the first part of E3. So they titrated it originally with just GDP. So that's exchange assay A, so this picture D. What they're doing is they have GDP tagged with this fluorescent group, and they're adding regular GDP without the tag to see if it'll be replaced. And as the fluorescence decreases, it shows the replacement of the tagged GDP with non-tagged GDP. So you can see that in blue is just just the G12C mutant without any compound added. Red with the up arrow is G12C bound to compound 8. And then red with a down arrow is G12C bound to compound 12. So in the previous test, compound 12 had 100% inhibition and compound 8 I think had 86, but they still are in that upper level of being able to target KRAS. So what it shows is there really isn't a difference between the mutant bound to 8 and the mutant bound to 12 when you're exchanging a fluorescent GDP with just regular GDP. It doesn't really show a preference. It's just switching one out with the other, so they all kind of happen at that same rate. Over here, however, they're titrating it with GTP, which is what turns RAS on. So here's kind of an illustration of that. So you have that fluorescent GDP bound, and then you're titrating it adding GTP, and you want to see if RAS will prefer to take the GTP and be on. This is where there's actually a difference. So G12C, once again, is in blue. That's bound to no compound. That is just mutated RAS doing its thing, and its thing is wanting to turn on. So it decreases in fluorescence quicker than the ones with the compound because it is not inhibited. It is easy to give it GTP, switch it out, and turn that compound on. It doesn't happen, obviously, instantaneously, but it does happen at a faster rate and an increase in concentration. However, you can see the difference between the red kind of decrease here and the slope over here. It's showing that these compounds 8 and 12 actually inhibit the switching from GDP to GTP because it takes more concentration of GTP in order to get the fluorescence to decrease, indicating that you have to pump more and more of that in there to finally get it to switch out with the tagged GDP. So this shows that it changes the preference of KRAS from GTP to GDP because it's a lot harder for these RAS bound to a compound to switch it out, which is good. And so this is a little more practical, actually getting into the pathway itself. So SOS is a GEF, which is what tells RAS basically to do its job and switch out GDP for GTP. It just exchanges the nucleotide. It doesn't necessarily have to be GTP though. Its job is just to exchange that guanosine-based nucleotide within RAS. So here's another illustration of what they're doing. It's basically the same setup, but they have that MANT tagged GDP, which fluoresces. And then they're either using EDTA, which if you remember from E1, is ethylene diamine tetracetic acid. It's a hexadentate ligand, and it is able to attack metal ions. So I think in E1 we used it with calcium, but obviously in RAS, the metal ion complex is magnesium. And the important thing about EDTA is that it's able to kind of take that magnesium ion away, which, like I mentioned earlier, decreases the tightness of the binding to GDP. So it just kind of lets it go, making it easier to switch out with another one. So that's what EDTA is used kind of as a control to show that if we take the metal ion away, of course it's going to switch out because it decreases that binding capacity. And then they introduce SOS itself, which is the activating protein in the pathway that turns RAS on and switches GDP out for GTP. So in this first pathway, in this first diagram, they just have G12C mutant, nothing bound to it. Intrinsic just means they didn't do anything. They added the RAS bound to the tagged GDP, they added GDP, and they just let it happen. So you can kind of see a decrease in fluorescence. So RAS did switch out the tagged GDP for regular GDP on its own, but not very efficiently and not very willingly because it didn't decrease at a steady rate. When you add sauce, it does decrease pretty steadily, which means that sauce is doing its job on the mutated RAS. So it's allowing that switch to happen from the tag GDP to regular GDP, which is what sauce does. And then EDTA, since it just snatches the metal ion away from RAS, obviously works really well because it decreases that coordination and allows it to easily exchange. So EDTA is just used to show that you can do this easily if you have like the right conditions. So the next is what they did when they had the KRAS mutant bound to compound 8, which is the vinyl sulfonamide compound. And you can see that the intrinsic, which is just doing nothing, letting RAS do its thing by itself, and the sauce are almost identical. This is showing that when you add this inhibitor, it prevents sauce from doing its job. It prevents it from exchanging that nucleotide. So if you think about this in a practical sense, if you have this inhibitor bound to a mutated KRAS and you have the pathway activated, then sauce is not able to exchange the GDP within RAS for GTP, and that signal is not able to be passed down through the rest of the pathway, which obviously stops that excessive proliferation. And then once again, you see EDTA, which goes in there, steals the metal ion, does its thing. The second is even more interesting because it's bound to 12, which had that 100% inhibition rate. It was the acrylamide. And the intrinsic value and the sauce are even at a lesser slope. It's almost vertical, meaning that it further inhibits sauce from doing its job and is unable to exchange that compound very well. Obviously, it still happens, but at a very slow rate. This is 15,000 seconds, and barely anything has changed, but EDTA is still out here messing it up. So this proves that it inhibits the function of the protein that's supposed to turn it on, and therefore cannot pass that signal down the rest of the pathway. The next part is finally getting into cell lines with cancer. So it's all fun and games, seeing if RAS is inhibited as itself, but does it actually work in a practical sense? So this is lack of coordination with RAF is what this is trying to show. So RAF is what RAS activates. So sauce turns on RAS, RAS activates RAF, and RAF kind of translates everything down the rest of the pathway. What they did here is they have two cancer cell lines of G12C mutants, H1792 and H358. In this top part, they just were able to get RAS out of the cell. So this like huge blob is apparently just RAS, but as you can see, it's not. And then they added the compound 12, which performed best in the previous, data. So at times 0 hours, 4 hours, and 24 hours, they took gels. So they're seeing if RAF is coordinated to RAS at this time with the introduction of the compound. So does it actually inhibit KRAS from tagging RAS and continuing the rest of the pathway? So as you can see at time 0, this basically means we haven't added any compound yet, and yes, RAS is coordinated with RAF. So when they removed RAS from the cell, you can still see BRAF in there. I don't know what's up with CRAF. You can kind of see a little bit of a dark line here, but the rest is kind of blurry. So I just chose to kind of omit that because BRAF is what you're really able to see throughout the rest of the gel. And then at time 4 hours, you see how different the band looks? It means that it successfully inhibited RAS from coordinating with RAF. So it stopped that transduction in the pathway. And then at 24 hours, it looks even lighter. So it's not doing its purpose, and it's not doing its job anymore. It's not sending that signal down to indicate like more gene transcription. It did not work as well on cell line H358. It didn't really say why. It actually said in the paper that it worked just not as well, but I don't think it really worked at all because you can still see dark bands of BRAF being coordinated to RAS even after 24 hours of the introduction of the inhibitory compound. So maybe it just works better on one cell line than the other. They didn't really specify why. And then if you go down here, WCL means whole cell lysate, which what I assume to mean is they just cut the cell open and let everything come out. So you have RAS down here for H1792 and 358. You have CRAF and you have BRAF. So even with the introduction of the compound, you're not going to get these to go away because everything was released from the cell. So the reason they put this in there is that, yes, BRAF and CRAF are in the cell. It's not like we removed them just to show this data and that it works. So they showed that these compounds, these proteins, are in the cell. However, if you add our compound, it works against stopping that coordination with BRAF down the further pathway. So this just kind of proves their point by showing that they are in fact present, they're just no longer coordinated to RAS with the introduction of compound 12. And finally, just another point on efficacy against cancer is another G12C mutant selectivity. So they introduced more cell lines. H1792 and H358, we know from the previous slide, they are lung cancer lines with G12C mutants. And then these other ones, CALU1 and H23, are also just cell lines that have G12C RAS mutants. And then they introduced some KRAS cell lines without G12C mutations. So wild type cell lines that do not have this particular mutation they're targeting. And they wanted to show that it, once again, only targets cell lines with that particular mutation. So once again, they used compound 12. And viability just means whether or not the cells survive. So what they're showing here is the induction of apoptosis, which isn't always an indication as to whether or not a therapy is effective. Sometimes you don't want to kill the cell. Sometimes you just want to inhibit that certain protein from functioning. But in this case, it did induce apoptosis in these certain cell lines. So as you can see, these colors are kind of hard to discern because they're all overlapping. But bright red, this maroon, this darker burgundy color, and the black are all the ones that have the G12C mutation. And then these tones of blue do not. So H1792, once again, we're proving that compound 12 is very effective against this particular cell line. It's slightly effective against 358, and then the other two, not so much. But you can see how drastic this decrease in viability is with the increase in concentration of compound 12. It is actually causing the cell to basically kill itself because it's no longer functioning. It's no longer sending that signal downstream. So this proves that it is selective for G12C mutations, but it's not really the best at showing that because even some of the ones that are mutated, this black line right here, still doesn't show a huge decrease in viability, but it is very effective against 358 and especially 1792. This bar graph over here shows the induction of apoptosis, so how frequently it happened within these cell lines. It just gives you a different way of looking at the data. So 1792, 358, CALU1, and H23 are all the ones that have the mutation, and then these darker boxes are the ones that do not. And you can see there's a drastic difference in the induction of apoptosis between ones that have G12C mutations and ones that do not. With the exception of 358, it's just messing up the data. But 1792, about a 34% induction of apoptosis, which, isn't a lot, but compared to the ones that do not have the mutation, indicating that the compound did not target that particular protein, it shows that there is selectivity amongst that G12C mutation versus regular wild type RAS, which kind of furthers their point that it is specifically designed for this protein that has this one mutation. And that's that.
All right. Nice job, Beth.
Question?
Does anybody have any questions? I guess the one thing that I think I can sort of notice is that, like a lot of your papers, actually probably mostly similar to Naya's, is this very slow and deliberate stepwise convincing process, right? Where they're sort of saying, okay, we have a compound that can attach to RAS, and it's dependent on the mutant. If that's true, then this other thing should be true, and then they show that. And then they make the system a little bit more complicated, and then this other thing should be true, like it shouldn't associate or bind to RAF anymore, and then they show that. And then they show that cells that have the mutation are uniquely sensitive to the compound. So that kind of like gradual unfolding is um, is sort of very common, I